Hi everyone, my name is Samir Abhiratna and I am from the University of Texas at Dallas. In this video, I am going to be talking about my research titled MavN IroT is a Legionella transmembrane ferrocyan transporter, metal selectivity and translocation kinetics revealed by in vitro real-time transport. First, let us know a little bit about Legionella pneumophila and Legionnaires disease. Legionnaires disease is the bacterial form of pneumonia. Nearly about 18,000 people are infected by the bacteria annually in the US and about 10% of them has the risk of death. Currently, the main treatment method for the disease is antibiotics. However, the problem of using antibiotics is occurrence of bacterial resistance. Therefore, efficient targeting the pathogen is needed to cure the disease. This cartoon explains the growth cycle of Legionella after infection in the host cell. After the infection, growth and the replication of Legionella is taken place in a host-driven vacuole called Legionella-containing vacuole or LCV. By doing this, Legionella can prevent being destroyed by the immune system of the host cell. However, the problem here is Legionella has limited access to the nutrients from the host cell. Legionella has a very interesting mechanism for nutrition trafficking from the host cell. It extrudes proteins called effector proteins by a type 4 secretion mechanism. MavN IroT is also one of the effectors which is inserted into the host-driven vacuole membrane after the infection. Even though there are over 300 effector proteins have been identified, in vivo studies reveal that Legionella only lacking the single system MavN IroT is incapable of growing and replicating inside the host cells. Therefore, effectors like MavN IroT are critical virulence factors, hence are potential therapeutic targets. Our goal in this research is to identify molecular mechanisms that are required to allow ferrocyan sequestration from endogenous cell cytosolic pool and transport into Legionella containing vacuole. IroT is a putative ferrocyan transporter. Our first goal is to test whether it transports ferrocyan. If yes, its transport mechanism. That means involvement of any counter ion in the transport mechanism. Therefore, our specific aims were developing an in vitro ferrocyan transport assay to express, purify and reconstitute LP IroT in artificial phospholipid bilayers and to make small unilamellar vesicles called proteoliposomes. And also to develop a tool to monitor real-time substrate transport and substrate selectivity. Also to determine molecular mechanisms of ferrocyan transport, which means uniporter, symporter and antiporter mechanism. Finally, to identify important residues and domains for substrate translocation. To achieve these goals, first IroT was expressed and purified recombinantly in BL21 competent cells and purified by immobilized metal affinity chromatography. Purity of the purified IroT was tested with SDS page gel and size exclusion chromatogram reveals the monodisperse size distribution. And the oligomeric state of the purified IroT was tested with SECMALS and SECMALS analysis reveals IroT is a monomer in the detergent micelles. Then purified IroT in detergent micelles was incorporated into proteoliposomes. In order to prepare lipid vesicles, commercially available E. coli polar lipid extract and phosphatidylcholine was used. Small unilamellar vesicles were prepared by freeze-thaw cycles followed by extrusion through 200 nanometer membrane filters. Then purified IroT protein in detergent micelles was incorporated into detergent destabilized liposomes. Detergent was then removed by binding to biobeads and final proteoliposomes were prepared. The directionality of incorporated IroT was tested by Western blotting by testing the accessibility of C-terminal histone. Control liposomes were also prepared following the same protocol without the protein. The vesicle size of prepared proteoliposomes and control liposomes were measured below 200 nanometer by DLA. This is essential to make unilamellar vesicles. SDS page analysis reveals almost all IroT is incorporated into proteoliposomes. IroT was incorporated into proteoliposomes with 50-50 right side in and inside out orientation. To monitor transporter-mediated real-time metal transport across the phospholipid bilayer in proteoliposomes, we selected turn-on type fluorescence probe fluorescein 3 inside the proteoliposome lumen. Fluorescein 3 is generated for zinc plus 2 ion, though it is not specific for zinc plus 2 ion. It gave turn-on fluorescence with all the divalent metal ions that we wanted to test. Also, metal ion titrations reveal that fluorescein 3 exhibits a linear response for all the metals that we wanted to test. Therefore, fluorescein 3 was used to measure the real-time metal transport events in IroT incorporated proteoliposomes. This is the experimental setup that we use to measure real-time transporter-mediated metal ion transport across the phospholipid bilayer. Fluorescence probe fluorescein 3 is encapsulated inside the proteoliposome lumen. When the metal transport is initiated with corresponding metal ion solution, and if IroT is transporting the metal into the lumen, metal ions binds to fluorescein 3 and gives a turn on fluorescence, and fluorescence response goes to saturation when the transporter reaches to equilibrium. This is the result for ferrous ion transport event. You can see when increasing the ferrous ion concentration, fluorescence response also increases and exhibits saturation kinetics, which is a typical for solute carrier transporters. Therefore, IroT is also a solute carrier transporter. Michaelis-Menten curve fit of ferrous ion transport kinetics turns out that the Km of ferrous ion transport is in low micromolar range, and IroT transports 10 ferrous ions per second per molecule of IroT. This turns out that IroT utilizes a transport mechanism involving conformational changes, and IroT is a solute carrier-like transporter and not a channel-like facilitator. Next, we wanted to test the transport mechanism. That is, whether IroT functions as a uniporter, symporter, or an antiporter. The LCV features a luminal acidic environment of about pH 6.1, which could be potentially exploited by IroT to drive ferrous ion transport. To test this hypothesis, we encapsulated pH-sensitive fluorescence dye pyranine inside the proteoliposome lumen and tested the fluorescence change during the ferrous ion transport. We observed an increase in pH in the proteoliposome lumen, which suggests that proton counter transport during the ferrous ion transport. Our results suggest that LP-IroT is a ferrous ion proton antiporter and it is a secondary active transporter. Next, transport selectivity of different divalent metal ions of first and second row transition metals were tested. Results turns out that IroT is a highly selective ferrous ion transporter over other first and second row divalent metal ions. In order to identify important residues for ferrous ion recognition and translocation, we generated a series of alanine mutants of potential metal coordinating residues. In the transmembrane region, we identified potential essential residues in the proximity of host cytosol membrane interface, or deeply embedded in the lipid bilayer, or located at the membrane vacuole lumen interface. We also mutated residues in loop 7 which diminish the in vivo virulence of Legionella in host cells. The C-terminal long soluble domain is also rich in potential metal coordinating residues, so we generated a delta C-term mutant to test the involvement of the C-terminal soluble domain for in vitro ferrous ion transport. These are the effects of mutations in the transmembrane region for ferrous ion translocation. We identified 3 mutants in which apparent Km was significantly affected while Vmax is similar to wild type LP-IroT, which turns out that those residues are critical for high affinity metal binding. On the other hand, we identified 3 residues which abolish the ferrous ion transport, and the protein becomes almost non-functional, and it turns out that those residues are critical for ferrous ion translocation across the phospholipid bilayer. These are the results of mutants in soluble domains. Mutations of potential metal coordinating residues in loop 7, which exhibited significant in vivo growth arrest of Legionella in host cells, had no effect for in vitro ferrous ion transport across the phospholipid bilayer. Therefore, those residues may be important for in vivo activity, possibly by protein-protein interaction or substrate transfer to unrecognized protein. However, for the delta C-term mutant, ferrous ion translocation across the phospholipid bilayer was significantly affected. Therefore, we can assume that C-terminal domain is critical to guarantee high capacity ferrous ion translocation. To measure the apparent dissociation constant for ferrous ion for wild type and delta C-term IroT, ligand competition experiments were conducted with MAGFURA2 and UV visible spectra was measured. MAGFURA2 could more successful when competing with delta C-term mutant than wild type IroT. Therefore, C-terminal domain is critical to guarantee high capacity translocation. Using a graphical method, apparent dissociation constant of ferrous ion for both wild type and delta C-term IroT were calculated, and apparent Kd for wild type IroT is 2.9 micromolar, which is in the same range as Km for ferrous ion transport, and the apparent dissociation constant for delta C-term LP-IroT is greater than that of wild type LP-IroT. These results turns out that C-terminal domain is critical to guarantee high capacity ferrous ion translocation, and also is involved in Fe+2 sequestration from the host cytosolic labile ion pool and delivery to the transmembrane domains. In summary, by developing a fluorescent in vitro platform for functional reconstitution in lipid bilayers, we established that MavN IroT is a family of ferrous ion transporters located in host pathogen-containing vacuole interface of Legionella, and it is a high affinity, high capacity and high selective ferrous ion transporter. And also we could establish the mechanism of ferrous ion translocation, that is IroT is a proton mediated secondary active transporter. And also we could identify putative ferrous ion binding sites responsible for ferrous ion transport. And we believe that our platform will allow to study other ferrous ion transporters in other families as well. That concludes my presentation, and thank you very much for watching this video. Link for the research paper related to this work can be found in the description box below, and this is my email address. If you have any questions, feel free to contact me. Thank you.